Okay, we've looked at carbohydrates and lipids, and now we're going to look at nucleic acids and proteins. And there's a close relationship between nucleic acids and proteins, as you'll see. And that's why we're going to look at them together over the next 5 slides. So nucleic acids get their name from their association with the cell nucleus. In other words, nucleic acids like DNA is found in the nucleus, and RNA comes from the nucleus, from the DNA in the nucleus. So they're associated with the nucleus, and that's why they're called nucleic. The acid part comes from the fact that when you put nucleic acids into aqueous solution, in other words, dissolve a nucleic acid in water, it will cause the pH of that solution to decrease, become more acidic. Therefore, nucleic acids are acids. Other than carbon and hydrogen and oxygen, there's phosphorus in this phosphate group and nitrogen in the nitrogenous bases that are found in nucleic acids. The monomers of nucleic acids are known as nucleotides, so what we're looking at right here is one nucleotide. Nucleotides are not all the same. So there are different nucleotides. This part of the nucleotide is the same. The phosphate group and the 5-carbon sugar is the same in each nucleotide. In DNA, the sugar is different in RNA. We'll come to that in a minute. But this is really what makes different nucleotides different, the nitrogenous base. There are 4 different nitrogenous bases in DNA, and there's a 5th nitrogenous base that is involved in RNA. But again, we'll come back to that idea. But that's what makes different nucleic acids or different nucleotides different. But if you're asked for the monomer of a nucleotide, you want to answer. If you're asked for the monomer of a nucleic acid, you want to answer nucleotide. The nucleotide is the monomer of nucleic acids, and you should be able to answer that question for all these organic biological compounds. What's the monomer, and what's the polymer? And in the case of nucleic acids, the polymer is a polynucleotide. So this represents a polynucleotide. When you link nucleotides together, you get many nucleotides, polynucleotide. This is another representation of a polynucleotide. The 3 compounds bound in one nucleotide are a sugar, a phosphate, and a nitrogenous base. And you can see that here from this nucleotide. The sugar, the phosphate group, and a nitrogenous base. And these are compounds. The phosphate group is made up of phosphorus and oxygen. You can't tell from this diagram, but there's phosphorus and oxygen in the phosphate group. The 5-carbon sugar is a sugar. It's a carbohydrate, therefore it has CH2O as its general formula. Nitrogenous base has carbon and oxygen and hydrogen and nitrogen. So again, you can't tell from this, but these are 3 different compounds, 3 different individual molecules that will come together to form this thing that we call a nucleotide. That's the monomer of nucleic acids. In a polynucleotide, nucleotides are bound between the sugar and the phosphate. So the way that these get strung together is between the sugar and the phosphate. You'll notice that the bonds here between two nucleotides is between the sugar and the phosphate. So this is what we often refer to as the backbone of a nucleic acid. Sugar, phosphate, sugar, phosphate, sugar, phosphate. That makes up the backbone. What's different about each nucleotide, again, is the nitrogenous base. So you can see here that there are different nitrogenous bases. So what we're looking at on this slide are the 2 different kinds of, the primary 2 different kinds of nucleic acids. We've got DNA and RNA, and right off the bat you can see there's a difference between them. So the general function of nucleic acids is to store and replicate genetic information. That's the general function, primarily of DNA, to store and replicate genetic information and use that genetic information. It's the sequence, and obviously that's an important concept. It's the sequence of the different nucleotides that allows them to perform this function. In other words, it's the order of the different nucleotides that make up a polynucleotide that contains the information, or that stores the information. It's just like letters in a word. Different letters in a word spell out that word. Different different nucleotides in a polynucleotide spell out information. The two major classes are DNA and RNA. DNA and RNA are different in 3 different ways. DNA is 2 polynucleotides, whereas RNA is 1. So you can tell that from the diagram here. Two polynucleotides linked together. RNA is only 1 polynucleotide. The sugar in DNA is deoxyribose. You can't tell from this diagram, but the sugars making up the DNA diagram here is deoxyribose, and the sugar making up the RNA diagram, or the sugar making up RNA in this RNA diagram, is ribose. And that's where the names come from, right? DNA is deoxyribonucleic acid. Deoxyribo is referring to the deoxyribose, the sugar making up the nucleotides. And RNA stands for ribonucleic acid, the ribo referring to the ribose, the sugar making up the nucleotides, and ribonucleic acid, RNA. DNA has the nitrogen-based thymine, while RNA has uracil instead. So if you look at these letters, A, T, G, and C, from previous science classes you may have learned about DNA having these different nitrogenous bases and different nucleotides, adenine, thymine, guanine, cytosine. Notice that in the DNA diagram there is the T for thymine, but if you look at the RNA diagram there is no T, but there is U, and that's uracil. So uracil replaces thymine in RNA. And this was just an introduction to nucleic acids. We're going to be looking at nucleic acids in a whole nother unit in much greater detail. So this is what this chapter is about, is just simply introducing the different organic biological compounds. Nucleic acids are one of those. So this brings us to our last organic biological compound, proteins. So again, just to run through them, we've got carbohydrates and lipids and nucleic acids and proteins. Those are the 4 classes of organic biological compounds. And proteins are very important in living things because they, as you'll hear me say numerous times this whole year, proteins run the show. Proteins do everything. And the relationship that I mentioned about between DNA, or between nucleic acids and proteins, is that the information contained in nucleic acids codes for proteins. That information is the information for making proteins, and that's generally the only information that's contained in DNA or nucleic acids, is the information for making proteins. So that'll give you an indication of just how important proteins are in the function of living things. So when we look at the chemical makeup of proteins, they're made up of carbon and hydrogen and oxygen, just like all these organic compounds are. But there's also nitrogen, right? So it's right there. Every protein has nitrogen because it's found right here in the monomers that make up the proteins. The monomers of proteins are amino acids. So that's what this represents, the general structure of one amino acid. And there are 20 different ones. There are 20 different amino acids. Can't tell from this diagram, it's just showing amino acids strung together to make a polynucleotide, but there are 20 different kinds of nucleic acids. So different proteins are different depending on what amino acids are strung together to make them up. That's what makes them different. The basis of this monomer name is that each amino acid has an amino group at one end. That would be this. An amino group is nitrogen and hydrogen, and a carboxyl group at the other end. This is the carboxyl group, and the carboxyl group is acidic. So the amino in amino acid is obvious. It's because of the amino group. The acid part of the name amino acid is not as evident. We could have called these amino carboxyls, but we don't. We call them amino acids because the carboxyl group is acidic. In other words, if you dissolve amino acids in water, they're going to decrease the pH of that water, or decrease the pH of that aqueous solution, which means increase the hydrogen ion concentration, right? Remember that inverse relationship. You decrease the pH, It increases the hydrogen ion concentration. Keep that straight. But anyway, that's why we call amino acids amino acids. All amino acids have an amino group and a carboxyl group. What's different between amino acids is what's in the diagram here is this R group. R. And so, again, there are 20 different amino acids, therefore there are 20 different R groups. And here we're looking at just two examples of two different amino acids, alanine and serine. And you can see that the only difference between them, the amino group and the carboxyl group is the same. This carbon, this hydrogen is the same, but the R group is slightly different. The R group in alanine is missing an oxygen, serine has. But that's the only difference, but that makes a difference. That makes a difference in the function of the protein that contains serine as opposed to alanine. The kind of bond that joins amino acids together is called a peptide bond. So these lines in between the amino acids here, you might even want to go ahead and label on the diagram, is a peptide bond. And because it's a peptide bond, we call amino acids, when they get strung together into the polymer of proteins, or polymer of proteins, is known as a polypeptide. So it's kind of named after the bond. Instead of being named after the monomer, it's named after the bond that links the monomers together. So this is a polypeptide. Proteins are the biggest and most complex of the organic biological compounds. So they're huge molecules compared to others. DNA can be really long, but proteins are just massive. They tend to fold up and tangle up into these big globular kind of molecules. Speaking of a big globular molecule, here we're looking at one. So this diagram is representing a protein. And it looks like this protein is made up of actually two different polypeptides put together, the red and the blue. And they don't identify what this protein is, but it looks very similar to me to a protein, an enzyme known as lysozyme that helps protect you from bacteria like getting into your eyes. Lysozyme is a component of your tears, and it helps break down the cell walls of bacteria that get into your eyes. That's lys means to break, so lysozyme is an enzyme that breaks. But anyway, a lot of the important function of proteins is in enzymes, so that's why I'm pointing that out. But you'll notice that we're zooming in on this small area of this big, huge protein, just to show that the polypeptide that makes up the protein is made up of amino acids that are strung together. So each one of these little spheres is an amino acid, and these are peptide bonds between the amino acids, and it's the sequence, very important word, it's the sequence of amino acids that causes the polypeptide to fold up in a particular way to give it its shape, which gives it its function. So there's this relationship between the sequence of amino acids that causes the polypeptide to fold and take on a particular shape, and it's the shape that gives the protein its function. And that sequence of amino acids comes from DNA. I mentioned previously the relationship between DNA and proteins. DNA, the information stored and copied in DNA, is the information of the sequence of amino acids to make proteins. It's the sequence of nucleotides in DNA that spells out the sequence of amino acids in proteins. I'll say that one more time because it's really important. It's the sequence of nucleotides in DNA that spells out the sequence of amino acids in proteins and makes every different protein different. All right, so that's what this says. So I'm not going to read this to you. So proteins, like I said, they run the show. They do everything. So we're just going to list here the different functions of proteins, and this is a relatively short list. We're looking at the just the major general functions of proteins. So there are structural proteins, like for example, primarily what makes up your bones and your muscles is protein. That's why, when you eat meat, you're eating the muscle of an animal, and you get a lot of protein from that, as you know, nutritionally. and that's because it's mostly made up of protein. Movement or transport, in other words, muscle contraction. So a lot of the protein that makes up the muscles is all about contracting, right? So that you can move your arm or move your leg or move your body. So movement, transport, like across membranes, like across cell membranes is what that's referring to. And there are different proteins that are involved in that. Signaling, same thing on the plasma membrane of cells. There are these things called receptors. And so that's how your nervous system works. That's how your endocrine system works. In other words, hormones. So there are proteins that are involved in all that. But it's all about these protein receptors that are in the plasma membrane or on the plasma membrane of your cells. That's how that works. Immunity. You've probably heard of antibodies. Your immune system, certain cells of your immune system produce these proteins called antibodies. And they are proteins that are designed to glom onto foreign invading cells or foreign invading viruses. I couldn't think of the word virus. It's a senior moment, I guess, huh? So that's how they function in immunity. Enzymes, they run metabolism. And this is arguably the most important function of proteins, enzymes. And this relates to the lab that we're going to do this week. We're going to do an enzyme lab looking at a particular enzyme, and it turns out to be happens to be the first enzyme ever discovered, called catalase. Catalase is an enzyme that breaks down hydrogen peroxide into water and oxygen. So it takes hydrogen peroxide, which is a toxin, and breaks it down into hydrogen and oxygen. And it's found pretty much in every tissue in your whole body, because your metabolism makes hydrogen peroxide, and that hydrogen peroxide needs to be broken down so it doesn't poison your system, basically, because it is toxic. So those are the general functions of proteins, enzymes being a very important one. So this variety of function is possible because proteins take on an infinite variety of shapes, and we call the shapes of proteins conformations. So if you see that word conformation, the conformation of a protein is simply a fancy word for the shape of a protein, and it's the shape of the protein that gives it its function. So it turns out we can identify 4 different levels of protein structure. And the first level is known as the primary structure of the protein, and that is the actual sequence of amino acids in that polypeptide. So this is a black and white diagram, grayscale diagram. So we're not seeing, they could be representing different amino acids here with different colors, but it's a much more simple diagram than that. But you got to remember that there are 20 different amino acids, and so the amino acids they're showing in this polypeptide could be any one of those 20 in any sequence. And as I mentioned on the previous slide, there's basically an infinite number of different sequences that those 20 different amino acids could be in, just like there's an infinite number of sequences that the letters of the alphabet, the 26 letters of the alphabet, can be put into to spell out whatever information needs to be spelled out. So that's the primary structure. The secondary structure is, again, how the amino acid sequence in a polypeptide causes the polypeptide to fold. And you'll find in different parts of a polypeptide different folding. So, one part of a polypeptide might fold in one way, while another part of a polypeptide folds in another way. And so one of the folding is known as a pleated sheet, and another kind of folding is known as an alpha helix. So different parts of polypeptides are going to fold in different ways depending on the amino acid sequence of that part of the polypeptide. We call that a domain. So one domain of a polypeptide might fold one way, while another domain folds another way. Finally, we get the whole polypeptide. So here we're just looking at a part of the polypeptide, but here we're looking at the entire polypeptide once it is done, and we can see those different domains folding in different ways. There's the pleated sheet, and there's the alpha helix. But the entire polypeptide is going to fold into a particular conformation, and that's known as its tertiary structure. The final 3-dimensional structure of the entire polypeptide, one entire polypeptide. That's the tertiary structure. But then some proteins, not all, will have a quaternary structure. So this one polypeptide all by itself that has this tertiary structure may carry out its function all by itself. So individual polypeptides can be a complete protein and carry out a function. Other proteins, however, are made up of more than one polypeptide that then come together or join together or bind together to carry out a particular function. And in this example, the quaternary structure is made up of two different polypeptides that have joined together. We don't know what this protein is. It's just an example. So the shape of the protein is known as its conformation. And so the first thing that happens is to string the amino acids together, and then it's a sequence of amino acids that causes the protein to fold up into a particular conformation. The conformation of a protein is based on the amino acid sequence. It's all about the sequence. It's all about the sequence of amino acids, and I'll remind you that the sequence of amino acids comes from the sequence of nucleotides in DNA. One polypeptide can function as a complete protein, like here at the tertiary structure level. So not all proteins go on to the quaternary structure level. Some stop here and function as one single polypeptide. Some proteins are made up of more than one polypeptide, each considered a subunit of that protein. So this example protein is made up of two different subunits, and you might want to go ahead and add that label, like you call this subunit A and subunit B or something like that. And each subunit is one separate polypeptide. You'll notice that the polypeptide they're showing here in light gray is this same polypeptide that we have here. This one came from somewhere else. It has a different amino acid sequence and therefore has folded up in a different conformation, but it makes up the other subunit of this quaternary protein. Hemoglobin, for example, the oxygen-carrying protein in red blood cells, is composed of 4 polypeptide subunits. So here we're seeing only 2. Hemoglobin would have 2 more. 2 of them are the same. It's called alpha hemoglobin, and the other 2 are called beta hemoglobin. So there are 2 of the same polypeptides, and then 2 of the same different polypeptides, making up the quaternary structure of hemoglobin. That's just one example. So again, one of the major and most important, arguably, functions of proteins is as enzymes in running metabolism, running the show, causing specific chemical reactions to take place in living things, and that's what we're going to be looking at next. We'll be looking at chemical reactions and how enzymes are involved in regulating chemical reactions.